Mitosis. Somatic cells and stem cells divide by mitosis. The mother cell, which undergoes cell division, is 2n, that is, diploid, which means the chromosomes present in the nucleus are in pairs. Before cell division, chromosomes are duplicated and their numbers are doubled up to 4n. The whole process of mitosis is completed through two main steps: karyokinesis, that is nuclear division, and cytokinesis, that is cytoplasmic division. The division of nucleus, that is karyokinesis, is completed through four steps: prophase, metaphase, anaphase, and telophase. Let us learn about them one by one. Prophase. In prophase, condensation of thin thread-like chromosomes starts. Due to this, they become short and thick, and they start to appear along with their pairs of sister chromatids. Centrioles duplicate, and each centriole moves towards the opposite poles of the cell. Simultaneously, nuclear membrane and nucleolus start to disappear. Metaphase. Nuclear membrane completely disappears before the beginning of metaphase. Chromosomes complete their condensation and become clearly visible along with sister chromatids. In metaphase, all chromosomes start arranging themselves parallel to equatorial plane, that is, central plane of the cell. Special type of flexible protein fibers, also called spindle fibers, are formed between centromere of each chromosome and both centrioles. Anaphase. In anaphase, centromeres split and thereby sister chromatids of each chromosome separate and are pulled apart in opposite directions with the help of spindle fibers. These separated sister chromatids are called daughter chromosomes. When each set of chromosomes reach at two opposite poles of the cell, they appear like a bunch of bananas. Telophase. The chromosomes which have reached the opposite poles of the cell now start to decondense. Due to this, each of the chromosomes again become like a fine, delicate thread and start disappearing. Very soon they are invisible. Nuclear membrane reappears around each set of chromosomes at both poles. Finally, two daughter nuclei are formed in a cell. Nucleolus also reappears in each daughter nuclei. Spindle fibers completely disappear. By the end of telophase, karyokinesis is complete, resulting in the formation of two nucleus with same number of chromosomes. Now the second step of mitosis, that is cytokinesis, begins. Cytokinesis. In cytokinesis, a notch is formed at the equatorial plane of the cell, which deepens gradually towards the center, and finally two new cells are formed. These newly formed cells are called daughter cells. However, in case of plant cells, instead of a notch, a cell plate is formed exactly at the midline of the cell, which grows towards the periphery, and thus cytokinesis is completed. Meiosis. Meiosis occurs only in germ cells. The process of gamete production and spore formation occurs by meiosis. After meiosis, one diploid cell gives four haploid cells. Meiosis is completed through two stages: meiosis 1, meiosis 2. Meiosis 1. This process completes in several steps. During this cell division, crossing over occurs between the homologous chromosomes present in the 2n, that is diploid mother cell. Homologous chromosomes are chromosomal pairs that are similar in length, gene pair, and centromere location. Crossing over results in genetic recombination. Thereafter, these chromosomes divide into two groups, and finally, two haploid daughter cells are formed, each with n number of chromosomes, which means Each cell possesses only one copy of a chromosomal pair. Meiosis 2. Meiosis 2 is just like mitosis. In this stage, the two haploid daughter cells formed in meiosis 1 undergo division, and finally, 4 haploid daughter cells are formed. Since there was genetic recombination in meiosis 1, all the 4 daughter cells formed are genetically different from mother cell and from each other. These cells now transform into gametes, which are always haploid, that is, have n number of chromosomes. After learning about cell division, let us do an interesting activity to observe different steps of mitosis in onion plant. Activity: take a medium-sized onion and keep it in a glass jar filled with water in such a way that the roots of onion remain dipped in the water. New roots develop within 4 to 5 days. Cut the tips of these roots and put them in a petri dish. Add few drops of iodine solution. Take one root tip on a glass slide and press it with the help of forceps. Add 1 to 2 drops of water on it and cover it with a cover slip. Observe the prepared slide under the compound microscope. Which step of mitosis do you see? We see metaphase step in the cell of the root tip. How did you identify the step? As all the chromosomes are arranged parallel to the equatorial plane of the cell, we can say that the cell observed is in metaphase step. Sketch the figure in your notebook.